Welcome to Genomics in Java, a great place for conversations over our favorite cup. This morning, we're joined by Dr. Kankshita Swaminathan, faculty investigator here at the HudsonAlpha Institute for Biotechnology. So thank you so much for joining us this morning. We're going to start with an easy question. What's in your favorite cup?
So it depends on the day. So it's usually tea, and it's whatever my husband makes in the morning, actually. This morning, it's a mixture of black tea with a bunch of spices in it.
That sounds delicious. I think I need to come to your house for tea in the morning.
You're welcome.
I will definitely have to join you. And what type of work does your lab do? What are the general interests in your lab?
So what we work on are grasses. So we work on a bunch of grasses that are useful for bioenergy, and some of them are multitaskers. So they are eaten as food, they could provide fuel, they're used as feed. For example, is something called sorghum, which is a cereal crop. We also work on a big tall grass called miscanthus, and many of you probably know it because in a lot of gardens, people use it for landscaping. It's a very pretty ornamental grass. It's also a very dense grass, so you can grow a lot of it very densely, and it's a perennial crop, and so it's being investigated as a bioenergy option. What am I interested in specifically is to actually understand the biology of these plants and the biology that would help grow these things in a sustainable way. For example, with perennial crops, you kind of put them in the ground and they stay for years together. So they go through the entire summer season, they grow up really big and tall, and then they senesce, which basically means they die down, the leaves go brown. And when they're doing that, they're taking whatever nutrients they took from the soil up into the leaves, and then from the leaves they are now moving it back into the next generation, which is the next year. And this could be seeds, which in some of these grasses are really tiny. In other grasses it can be big, and then they have something called a rhizome, which is the rhizome most people would have come across is like a ginger. Mm-hmm. Right? And so this stays underground, and it's nothing but a modified stem. So it's the same stem, and underground it kind of changes its morphology, which means it changes the way it looks, and it starts becoming a storage organ. And so at the end of the year, through winter, all the reserves go down there. And then in summer, when the daylight changes and the temperatures get warmer, the plant grows up again and feeds the new shoots that come up. And so this cycling helps it conserve resources, right? So resources are not taken away. And so they kind of continuously, we call it remobilize and reusing resources. And like any organism, there are genes behind this. And so what I'm interested in is to understand the genes and the genetics and how does the plant actually do this. So that's a major question for us.
What an incredible set of information to be able to navigate through as we look at the use for plants for bioenergy, as we look at plants for crops, and we think about that sustainable agriculture. So recently, your lab worked on a publication on miscanthus. Can you tell us a little bit more about that publication and about the work that your lab did?
So that publication was a collaboration with a bunch of different groups. So one of the things when I said, we're interested in the genes involved in different processes, you actually have to know what those genes are, right? Like, so you need genomics, you need information to be able to explore. So for you to be able to connect, okay, I like the fact that this plant has big seeds and this plant doesn't. What's behind that? And so for that, you need a certain amount of genetic and genomic resources. And so this group basically was a collaboration between the main people were the University of Illinois in the US and the University of Berkeley, California. And so the group at Berkeley put together the actual genome. So which means that they took sequencing data that we get today. And then they kind of piece the jigsaw puzzle together to kind of recreate the entire chromosomes. And so you can do this computationally. And then there are computational methods which can then predict all the different genes in there, right? And then you can use experimental methods to confirm some of this. And at this point is where things start falling off. So we've gotten that far with just genomics. And so you can understand, what are the genes that are expressed in this particular organ versus the other. So what are the genes that are all showing up and therefore probably playing a role in the rhizome versus the stem, for example. And so we kind of collected data on all these things through this paper. So that's what's in the paper is I call it the data collection phase, where you now created a certain number of hypotheses to say, I know that there's this list of genes that appear to be interesting because they might do this. And that's where the bottleneck has been now, because in the last 10 years, genomics has just exploded and we've got so much information at hand. But what we still don't know is what do these genes do? Because then we're looking to say, this gene looks like this other gene, which in my model organism, which is like, something called Arabidopsis, which is a little mustard.
Little bitty.
Little tiny little mustard, very different from the crop plants that we work with. But a lot of work has been done on that because we can grow it in a little lab and we can grow it under, bad light conditions and things like that. And it has a very short life cycle. But it's far away. And so you're like, okay, it looks like that. So maybe that's playing this role, right? But it's just you're building stories and your evidence is kind of very rapidly declining. And that's where new technologies that are now available are helping. So we've got gene editing tools. We've got, better plant transformation methods, which basically means that you can now say, I think this gene does this. What happens if I take this gene out of the picture? So I mutate it so that it is no longer functional. What happens to the plant? Is the plant perfectly fine? Or does it actually have an effect on the plant? And does the rhizome, become smaller or bigger or, rots more, or what happens, right? So questions like that can now get answered. And so the foundation was laid by this paper, and that's kind of where now the next set of questions, that's where we're going.
Thinking about the field in general, where do you see the field going in the next 10 years using those tools that you mentioned, using all this technology that we have available? What are some of the promises and challenges that you see in agrogenomics?
Well, so I think for sure the new technologies have now opened up. Firstly, the foundation with genomics has now opened up a whole lot of questions, right? So now we've got information for a lot of crop species. We've got information to understand the diversity in these crop species. So we've got information on how different a gene can be in very related plants. And so using all this information now, we can build these stories and hypotheses, and using these new technologies where you can actually go into a plant and change something to really understand, because that's your proof of principle, right? And so to really understand it, then you now slowly build a catalog of genes and their functions, right? And so ultimately, I think the challenge in the next so many years is you've got a growing population, you need to feed and fuel the world, and we want to do it on the land we've already allocated, right? Or the land that's lying fallow. So we want to do all of this without actually destroying our forests and destroying other habitats, right? And so to do all of that, we need to really understand these plants. And we need to be able to understand, I think, this way of using resources efficiently so that we can pull together through breeding and, maybe biotechnology, you can pull together these plants which actually can be sustainable.
Thank you so much for the work that you do and for joining us today and talking with us. It was Such a pleasure to have you with us. And so until next time, stay curious.